everyone for joining the webinar today. I would like to talk to you about next-generation sequencing, the sources of error, and roles of replicates. I would also like to introduce you to my company, e.g.Quintiles, but before I do so, let me talk a little bit more about what we will be discussing today. I'd like to start off with a discussion of our paper that was recently published in Nature Reviews Genetics entitled The Role of Replicates in Error Mitigation in Next-Generation Sequencing. And in addressing that article, we'll talk about sources of experimental error and the role of replicates for addressing those sources in high-throughput sequencing, otherwise known as next-generation sequencing. And then we'll move on to talk about one approach that was outlined in the paper for replicate analysis. From there, I'll talk to you about other considerations for abating error, and these include the computational and configuration issues from which error may arise. And following on that, we'll talk about other approaches for mitigating these types of error and post-publication new developments. From there, I will finish up with discussing some real-world approaches to abating error so that you can see how these methods are actually applied at e.g.Quintiles and beyond. And then following that, we'll have some concluding remarks. So very briefly, I wanted to mention that e.g.Quintiles began its gene expression services, offering these services in 2001. And following this timeline, you can see that we began offering next-generation sequencing services in 2010 with our Illumina sequencing, and we also have other types of sequencing services available. So I would invite you to revisit the recorded webinar if you are interested in more details on this slide. I'm not going to go over the entire slide, but I would bring to your attention one bullet at the bottom right of the slide, which we are very proud of, which is the annual client retention rate that we see is in excess of 85% year over year, and we're very proud of that. We're very happy to be able to make our clients very happy, and they really enjoy our data. So without further ado, I'd like to begin a discussion of our paper that was published earlier this year in Nature Reviews Genetics entitled The Role of Replicates for Error Mitigation in Next-Generation Sequencing. And if you have not had the opportunity to download and review this paper, you can access it for free at the URL at the bottom of this slide, which, again, this is a recorded webinar, so I would invite you to return to the recording if you need to record that URL. All right, so let's start with the first portion of the paper, the sources of experimental error. Now, before we can talk about the sources of experimental error, it's important to understand what the different genetic variants of interest, what the different types are, because error can obfuscate the identification of both germline type variants and somatic type variants. So what is a germline variant? A germline variant are those specific alleles that are inherited by an individual. So these are the germline alleles that you'll see in high proportions throughout the individuals, and the individual's genotype you can be thought of as the average of those alleles. Somatic variation, on the other hand, also known as acquired mutation, is the alteration of the DNA that occurs after conception. So somatic variants frequently appear at lower rates than those germline or genotype variants, and somatic mutations play a very important part in oncology, in particular in tumorigenesis and also other disease states outside of oncology. So you can see that by improving germline variant detection, you can also improve the sensitivity of finding those true somatic variants, because once you have the germline variants, any variation that you detect outside of that would either be somatic or error type variation. Okay, so now that you have a clear understanding of those two variant types, then it becomes more clear why the Figure 1 caption reads as sources of unexpected or erroneous variants and the tools to cope with them, because once you've identified, as I said, the genotype, then what's remaining is the error, but also it's important to understand that you have the somatic variants in there as well. All right, so figure 1 can be divided into the top half, which addresses the experimental sources of these sequence variations, and the bottom half, which addresses and illustrates the computational steps. So for the purposes of discussing replicates, we would really like to focus on this top half because replicates are best at addressing stochastic type errors and less important for addressing the kinds of variation that you might see arise in the computational steps. And at this moment, I'd like to pause for a polling question. Andrew.
Great, thank you. We certainly do have a polling question here for the audience. And you can vote on this in real time by clicking on your screen. The question that we have for you, which step is the most problematic for quality control in your experiments? And your options are sample preparation, library preparation, sequencing or imaging, analysis, or you're uncertain. Again, that question, which step is the most problematic for quality control in your experiments? And again, you can vote on that in real time by clicking on your screen. It looks as though most of you have voted at this point, so I'm going to close polling now and share the results with you. There we have it, 32% of you said analysis, while 25% said library preparation, 23% said sample preparation, and 21% of you were uncertain. 0% of you said sequencing/imaging. And with that, I'll hand back the mic to you.